हाँ भाई ये देखना एक बार जो है ना मेरे पास वैसे है ना जब भी ये शेयर होगा ना ये वो लिंक कर देंगे उस टाइम बताना।
Hello, Professor Bell.
No. Hello. Hello, Richard.
How are you?
Very nice. Very nice to see you after a long time.
Yeah, you look very well.
Yeah.
That's great. Great to hear.
Yeah. Now I'm at a different place, and we have a good 7-star hospital here, multi-specialty hospital here. So maybe next time when you come to India, we will arrange a conference here now. It's a better place, much better place.
Where is that located?
It is near Chandigarh, and it's a A++ university, only in first 2% universities in India.
Fantastic.
Yeah, so I have joined here in research and development, and you see our director Virendra Pahil.
Yeah.
Good morning. Good morning, Dr. Richard. I am Dr. Pahil. I am Dean and Director of Agriculture.
Nice to meet you.
Okay, thank you.
Pooja Malik is the star for Women's Day. She is the anchor of the program today.
Good morning.
And, good morning, Pooja. He's saying good morning to you.
Yes, sir. Good morning.
What
And
time is it over there?
Professor D.P. Singh is president of the Society of Sustainable Agriculture, which organized the conference in Agra.
Thank you.
Yeah,
good morning. Good morning.
Yeah.
Thank you.
He was earlier Vice Chancellor of India's 4th biggest agricultural university, Jawaharlal Nehru Krishi Vishwavidyalaya at Jabalpur. And he is now even much bigger person. He is chairman of the National Agriculture Science Fund.
Wow. That's a very big job.
Yeah. Richard, you have not visited after 2019 India?
Yeah. I thought I would come along anyway.
Yeah, very good. So we wanted to invite you again, but due to COVID restrictions, we couldn't organize any conference. Yeah.
Yes, we haven't traveled.
Now we are planning.
Great. We look forward to coming back to your beautiful country.
Yes. Maybe David also.
Hello, Dr. Bhatia. Good morning.
Good morning, sir. Good morning. A very good morning to everyone. Good morning, Pooja. Good morning, Dr. Bell. Good morning, everyone.
Thank you.
Morning.
You know Dr. Bhatia, you know Dr. Pooja and Dr. Bell. I am Dr. V.S. Pahil. I am Director of Agriculture. मिलेंगे बैठेंगे आपके साथ.
Sure, sir. I look forward for the moment. I look forward to it.
We will plan a good project for international funding.
Definitely, sir. That shall be a great pleasure.
Richard is here, and the president of Opera is likely to come.
Yeah, Richard is here.
Richard, Professor Ruby Bhatia is our head of the gynae department.
Nice to meet you.
Good morning, Dr. Richard. Good morning.
Good morning. I look forward to meeting you in person one day.
David Olsen को भी करना है और उनको भी करना स्लो बोला.
ये कहाँ है?
Here he is. Good evening, David.
Yeah. हाँ जी.
Look as fresh as a daisy, David.
Yeah, it's a
Good
special
morning,
day.
Professor David.
David has also joined.
Yes, sir.
Very good, very good. तो जगदीप किसको ज्वाइन कराने गया? अच्छा, तो वो ज्वाइनिंग वहां से होगी क्या? है? पूजा ज्वाइनिंग उधर से होगी क्या? तो फिर वो यहाँ से जाता क्यों है? हाँ, कल कॉल कर उसे. उसको करा ज्वाइन कराया. वो उसको फिर दे दिया.
पूजा. हेलो पूजा.
हाँ जी सर.
कब स्टार्ट करना? कब स्टार्ट करना है?
After 5 minutes, sir.
कितने कर हो गए हैं? हाउ मेनी पार्टिसिपेंट्स?
अरे वो भी टाइम 10 AM का लिखा हुआ है ना उसपे, प्रोसेस से. वो गलती रहेगी ना मुझे. किसने चालू रखा था जगह? पूजा म्यूट करो.
हो रहा है अरे पूजा क्या हो रहा है? छोड़ क्यों मारे पूजा? हेलो पूजा. हेलो पूजा. हाँ? नहीं अब तो ठीक है. डॉक्टर पूजा Hello, Dr. Behel. Should we start now?
I will
पूजा
Yes, sir. Shall we start, sir?
शोर कैसा हो रहा है ये? अब ठीक है.
Kindly keep yourself mute, other participants. It's my humble request to other participants that kindly keep your mic off. Sir, shall we start? Professor Behel, shall we start?
David Olson joined. Hello. Welcome on board all the speakers, and I'm waiting for Professor David Olson to join and Sloboda to join so that we may start the greeting parts fast. Has Professor David Olson joined? Professor David Olson has joined?
Yes, he's here.
Ah, he is here. Very good. Thank you. So maybe we start now, and there was a small mistake in the announcement, and that has created a little bit of problem. But now we can start. Okay. The seminar was decided almost a month back when I talked to Professor David Olson about joining the hands for another activity, maybe in the form of conference or webinar, which he very eagerly and very nicely agreed. And he said that let us decide a common topic. So we decided a common topic, and he took over the responsibility of organizing speakers. And in fact, he is the real and in true sense today's chair of the webinar, but he, maybe because of the hot times in Canada, this time it is late night, so he opted to be off scene, but all credit goes to him to organize today's speakers for today's webinar. And we then thought that since it is international webinar, we should also have one speaker from India, so that the timings are in control. And that is how we identified Professor Ruby Bhatia to be the speaker from India. So with this background, I say that it's a Women's Day, so the anchor should be a woman. And therefore, I request Dr. Pooja Malay to be a hostess of today's webinar and anchor the whole proceedings of today's webinar. Dr. Pooja, please.
Yatra naryastu pujante, ramante tatra devta. Where women are honored, divinity blossoms there. It's a great pleasure for me to anchor today's important webinar. Our respected Professor Rishi Bahel organized 2 international conferences earlier in 2019 at Hisar and Agra, where Professor David Olson and Professor Richard Sefri delivered keynote lecture and sensitized international audience towards the optimal pregnancy environment risk assessment in relation to preterm birth. As nutrition is one of the deterministic factors for optimal pregnancy environment risk assessment and minimize the risk of preterm birth, promote proper fetus development and health of the neonatals. Therefore, agriculture comes into play. That is how the today's webinar has been conceptualized to celebrate International Women's Day dedicated to women and neonatal health. The webinar is jointly organized by OPERA, Optimal Pregnancy Environment Risk Assessment, DOHAD, Developmental Origins of Health and Disease, IFSADA, International Foundation for Sustainable Development in Africa and Asia, and SARM, Society for Sustainable Agriculture and Resource Management. Besides, MMDU Department of Agriculture and College of Medical Sciences. In this context, first of all, I invite Professor D.P. Singh, President SARM, to greet the mentors, speakers, and participants. I consider it as a privilege for me to introduce Professor D.P. Singh, currently the National Agriculture Research Fund Chair of ICAR and President SARM, India. Professor D.P. Singh is an agronomist by profession. He has been Vice Chancellor of Jawaharlal Nehru Krishi Vishwavidyalaya, Jabalpur, Member Farmers Commission, Bhopal, Consultant Farmers Commission, Haryana, Dean College of Agriculture, CCS Haryana Agriculture University, Hisar, member of many QRT teams of ICAR, member of various accreditation committees. He has won many national laurels and awards. I cordially invite him to greet the mentors, speakers, and participants on behalf of SARM and offer his valuable remarks on the importance of nutrition. Professor D.P. Singh, sir, kindly keep your mic on.
No,
on. Professor D.P. Singh, kindly do your mic.
Unmute. That's up. Kindly unmute your mic.
Am I audible now?
Yeah. Now you are audible. Yeah, please.
Thank you, Madam Pooja, for nice words. As you just told, the Society for Sustainable Agriculture and Resource Management was started in 1997 at Haryana Agriculture University, Hisar, India. And since then, Professor Bahl has been the secretary of the society and a very active member since the beginning till date. And for that purpose, we have also one of our branch in Göttingen with the collaboration of Professor Georges. That is the part of India to organize other conferences around Europe or so. And they have one facility there, though he will tell in detail, Mahatma Gandhi House in Göttingen, where his office is also located. We also share his office whenever there is conference or any other activity in Germany. So Germany and India, particularly the EFSADA and SARM, they are collaborating with each other whenever the collaboration is required. And in that context, India or SARM have organized large number of conferences in Europe, particularly in Germany, Czechoslovakia, Poland, Hungary, and other parts of the Europe, and also in Canada and China. So we have also the large number of conferences which were organized under the SARM in India, and the mega size conferences were organized in 2005 and 2007 at Jawaharlal Nehru Agriculture University, Jabalpur, India, where the persons from 7 countries were participated in those mega size conferences. And in that context, we had also, because most of the persons came with the family, also organized the flower show, because Madhya Pradesh is the hub of the flowers, and in that context, roses and other things. So most of the ladies, they participated in those flower shows. And in addition to the conferences, which covers most of the themes related to natural resource management, and that include also the areas like livestock and fisheries, which are also very important part of our sustainable agriculture. And we also support the students workshops for their exposure to different key areas, and also support their participation in forms of traveling grant within the country. The society also have its own fund and provide the seed money to everybody within the country or outside country on the returnable basis to organize any conferences or workshop within India or outside India. So in that context, Professor Bahal is a key person who is collaborating with the various key speakers, key institutes within India and outside. And I am happy that since then, because the society has already completed around 25 years. So we are together, though we are not at one place, because nowadays the network, other facilities are so developed that you are always near to us. So coming to the real topic, that we know that particularly in Asia and Africa and particularly in India, the large number of women, they are suffering from the various deficiencies of the micronutrients, proteins, and several other things. And in that context, the government of India has got very big support in terms during the particularly COVID period for 2 years for providing 5 kg rice and wheat, 1 kg pulse, 1 kg refined oil, 1 kg sugar, and 1 kg salt to 800 million people since last 2 years. And that is still continuing. And for that purpose, particularly, the women are major beneficiaries. And there is another program which is operating in India that is called Anganwadi. And in Anganwadi, that is the women organization at Panchayat level. So in that context, the children and the women, they are provided with iron tablets and other things, and also the proper guidance to the pregnant mother and also the growing children. So that part is again helping the women for their various benefits. And besides that, the plant breeders, particularly in India, they are putting a lot of efforts to develop the fortified varieties, which contains the vitamin A, iron, zinc, and other calcium, phosphorus, other things. And as you know, Dr. Swaminathan, who is the father of Green Revolution in India, he was also the chairman of National Farmer Commission. And in that context, he just requesting the government, because in India is also have, of course, coarse grains like sorghum, palm millet, maize, and small millets, which are very rich source of calcium, iron, phosphorus, vitamins, and other things. And the fortified varieties which have been developed by the breeders, that is your golden rice, which is a very rich source of vitamin A. Then we have also large number of varieties, or 3-4 varieties of wheat, which are rich in zinc and iron. And similarly, the millets varieties, they are also again rich in calcium, iron, zinc, and other micronutrients. So Professor Swaminathan is always emphasizing and giving the name of these coarse grains and millets and small millets as designated as nutritious food. So he wants that this should be also the part of public distribution food grain system. But the government, particularly the people, they prefer like the rice and wheat and pulses. So our government is emphasizing in the coming days or years to put this part, millets and small millets, as part of the public distribution system. Then the women and children, India is also hub of large number of medicinal plants, and in that context, one of our yoga guru, that is Ramdev, Swami Ramdev, he is also emphasizing particularly the use of several medicinal plants to increase the immunity among the women and children and other members of society. So I am not the person who is doing with the health also, as you know. So that is just my few achievement of Government of India and other scientists. Because Dr. Swaminathan is a very big person. He has been the food prize winner and also the father of Green Revolution. So he interacting all the time to take care of the pregnant mother and also growing children to have the healthy food in their diets. So with these words, I thank the organizer and particularly the Professor Well, who is still very active. We are also very active to organize such a type of the webinar during the COVID period. So thank you very much.
Thank you so much, Professor D.P. Singh. Thank you so much, Professor D.P. Singh, for your valuable remarks. I now cordially invite Dr. K.W. Georges for greetings and remarks on behalf of IFSADA and AASF, Mahatma Gandhi House, Germany. Dr. K.W. Georges is a native of Ethiopia and obtained his PhD in chemistry from Tübingen University, Germany. He has a special head and heart for sustainable development in Africa and Asia. He is currently president of Afro-Asian Studies Promotion Association at Mahatma Gandhi House, Göttingen, Germany. Now I call upon Dr. Georges for his precious remarks.
Thank you very much. I feel privileged to address this international webinar on women and neonatal health. I would like to, on behalf of my association, the Afro-Asian Studies Promotion and its subunit, the International Foundation for Sustainable Development in Africa and Asia, and myself, I send you greetings to America, India, Australia, Canada, from a small town in Germany called Göttingen. Belonging to our association, we have a students' hostel by the name of Mahatma Gandhi. Our association was founded in the late 50s, exactly in 1959, during the Cold War. Unfortunately, after 60 years, we currently live in a situation where war is raging in Ukraine. The name Mahatma Gandhi obliges us to raise awareness of peace and work for its achievement. Peace is appreciated when lost. Let us hope that rationality will prevail. We already know that the coronavirus has inflicted damages on human beings and the world economy. And now we are faced with a far more dangerous crisis, one that we deeply hope can be resolved. Since we established the International Foundation for Sustainable Development in Africa and Asia in 2007, we have organized several international conferences in different countries such as Hungary, Poland, India, and of course, India. We are fortunate to have Professor Richie Bale as a member of our association and as the coordinator of our global network. His contributions to the success of the International Foundation for Sustainable Development in Africa and Asia cannot be listed. Thank you for your commitment and your contributions. Beyond climate change and world peace, there are several topics that are directly related to the well-being of men and women. Such is the case with today's topic, which is held on Women's Day, a topic that specifically affects women who give birth and those who are born, be it female or male. This topic is not purely medical, but is multifaceted and surely will leave a lasting impression among the participants of this webinar. Also coming from an East African country, Ethiopia, and having in mind the comparison between Germany and my country, I can see and feel the importance of this topic to raise awareness for mothers, and provide tools for politicians and policymakers. In the invitation to this webinar, it was stated that 23% of preterm births occur in India, and I assume that this is proportionally similar to their population numbers in other countries. It becomes clear that the study of pregnancy becomes important to understand preterm birth and all the issues that revolve around pregnancy, which I think as a layman highly determine life after birth. I hope and trust that this Women's Day webinar will open our eyes to the topic of birthing mothers and their exposure to various risks during pregnancy. In our Ethiopian society, a lot of consideration is given during a woman's pregnancy, where the husband tries to fulfill all his wife's wishes, knowing that pregnancy is not an easy phase of life for women. To put it crassly, there is a saying in my country that a pregnant woman hovers between life and death. Finally, let me say a few words about our association. From March 20th, that is to say in 12 days, depending on the actual development of the corona front, many restrictions would fall, which has prevented us from organizing international conferences. Fortunately, corona has forced us to use Zoom, which gives us more possibilities for frequent webinars, which will continue to be held in addition to the international conferences. With that said, I wish this webinar to be a success. Thank you for your attention.
Dr. Pooja. Dr. Pooja. Dr. Pooja. Dr. Pooja.
I'm finished.
Dr. Georges has finished.
Okay. Thank you, Dr. Georges, for being with us and your valuable remarks. Now I am happy to invite Professor Virender Singh Pahil, Director, Department of Agriculture, MMDU, for the welcome address on behalf of MMDU Mulana and opening remarks about the role of women in agriculture.
Thank you, Dr. Pooja. Good morning, good evening, and namaskar to all of you. First of all, I wish you all a very happy International Women's Day, which we are celebrating by organizing this international webinar on women and neonatal health. So I welcome you all, the participants and the distinguished speakers throughout the globe on behalf of me and on behalf of Department of Agriculture, Maharishi Markandeshwar, Dean Toby University, Maulana Ambala in Haryana. I am also thankful to all the collaborators and the moderators. Although I am not an expert on the subject of today's webinar, however, being organizer of the webinar, I would like to discuss the role of women in agriculture very deeply through this small presentation. भाई देखना कहाँ से हो ये है और ये is it visible? Role of women in agriculture. Presentation is visible to all?
Yes.
Okay. So, our first Prime Minister, Pandit Jawaharlal Nehru, has rightly said, in order to awaken the people, it is the women who have to be awakened. Once she is on move, the family moves, the village moves, and ultimately the nation moves. As Dr. D.P. Singh talked about Dr. Swaminathan, he is a big scientist, he is father of agriculture in India. He has also said some historians believe that it was women who first domesticated crops and thereby initiated the art and science of farming. While men went out hunting in search of food, women started gathering seeds from the native flora and began cultivating those of interest from the point of view of food, feed, fodder, fiber, and fuel. So far as status worldwide is concerned, 70% of the women are living in poverty. Women do more than 60% of the hours of work throughout the world, but earn only 10% of the world's income and own only 1% of the world's property. The value of unremunerated work was estimated at about $16 billion, from which about $11 billion represented the invisible contribution of women. Women are paid 30 to 40% less than men for comparable work on an average. This is on an average basis. 60 to 80% of the food in most developing countries like India is produced by women. Women hold between 10 to 20, only 10 to 20% managerial and administrative jobs. Similarly, women makes up less than 5% of the world's head of the state. So this is the global situation of the women. Similarly, as far as status of women in India is concerned, in India about 49% of the total population are women. Literacy rate is only 54%. In rural women it is much less, not more than 32%. Urban women literacy rate is about 54%. And the share of female share on non-agricultural basis, with non-agricultural basis employment is only 17%. So participation of women in workforce is only 14% in urban sector, whereas in any Indian state, women and men are not equal in wages. Only 9% of the parliamentary seats are for women, less than 4% in High Courts and Supreme Court, and less than 3% administrators and managers are women in India. So this is the situation of women in India. So this is important. Why women are in agriculture? The first is in order to eradicate the poverty. The first is for eradicating the poverty, women's role is very important. To ensure food security. Food security is very important for India. So women's role is again very important. To increase their stake in agriculture, to increase economic contribution, and also to have ownership in land, livestock, etc. Again, the woman is very important, playing very important role in agriculture. Women are the really molder and builder of any nation's destiny. They play a significant role in any economy. They are regarded as backbone of the rural economy, and most of the women perform various types of work for their livelihood, and agriculture is considered as biggest unorganized sector where large number of rural women take part actively. So far as contribution in agriculture of women is concerned, they are responsible for household food and nutritional security, as Dr. D.P. Singh rightly said. 90% of the hoeing and weedings are done by the women for food production. 80% of the work on food storage and transports. More than 90% of post-harvest management, including food processing, providing water and energy, are done by the women. More than 60% of harvesting and marketing is also done by women. So women are doing this. Women have multi-dimensional role. In agriculture, they are doing sowing, transplanting, weeding, irrigation, fertilizer application, plant protection, spraying, of the pesticides, harvesting, winnowing, storing, etc. So far as domestic work is concerned, all women do cooking, child rearing, water collection, fuel, wood gathering, and household maintenance. So far as allied activities are concerned, cattle management, the women are looking after the cattle, they are collecting fodders for cattle and doing milking, etc. Horticulture, vegetable production, flower production, fruit production, all are done by women. So in sericulture also, silkworm rearing and cocoon production, all are done by the women. So here are some of the highlights of the work of the women, as they are the major force in the agriculture. As they are just taking milking, and they are doing other domestic works for animals also. They are doing good work. Similarly, they are doing this plant protection activities, spraying in the field, harvesting, and just for rearing the animals as well. Just to conclude, women contribute to two-thirds of the world's work hours. Women produce 50% of the world's food supplies. Women work in field, take care of families, and manage household. Despite the services rendered by women in family and workplace, they make up for nearly 70% of world's poor and more than 65% of the literate. So in true sense, women are the unsung warrior, especially of Indian agriculture. Thank you so much. In the last, I hope that this webinar will be very useful for all of you. I wish the webinar a grand success. Thank you, one and all. Thank you so much.
Thank you, sir, for your precious remarks about this webinar. Dr. Deborah Sloboda is a professor and chair of research in the Department of Biochemistry and Biomedical Sciences at McMaster University. She holds a Tier 2 Canada Research Chair in Perinatal Programming. Dr. Sloboda's laboratory investigates early life impacts on maternal, fetal, and placental developmental, and how this mediates the risk of non-communicable disease later in life. She represents society named as DOHAD. I cordially invite her to offer greetings and remarks on behalf of DOHAD.
Hello, everyone, and happy International Women's Day. I'll thank you very much for the opportunity of speaking on behalf of the International DOHAD Society. I will just try and share my slides. I only have really 2 minutes of words to say. Can you still hear me?
Yes, ma'am, you are audible.
Okay, great. I'll see if I can get this to work here. Sorry, my computer is not cooperating. So perhaps, perhaps instead of waiting for the slides, I'll just speak. The International DOHAD Society is a society of many members from across the world that it comprises members of over 60 countries, many of whom reside in India. And I hope some of the members might be watching right now. I wanted to say that the International Society will also have an International Women's Day webinar tomorrow, if you'd also like to join us later. The society, sorry, I'm really having trouble. Can you still hear me? Because I have a big lag on my end.
I can still hear you, Deb.
Okay, great. Sorry about that. It seems maybe the internet doesn't work at night in Canada. So anyway, I'll repeat that the International Society is made up of members from over 60 countries from around the world. We have many members comprised of individuals who are very interested in agriculture specifically. And I will invite anyone who is interested in wanting to know more about the early life environment and the role that women and pregnant women contribute to the health and well-being of the next generation to consider joining the DOHAD Society. We have monthly worldwide webinars that you are welcome to join. For now, these worldwide webinars are free. And they occur on the first Wednesday of every month. And we hear from all sorts of speakers from all over the world on various topics, including maternal and child health. We also have a journal of the developmental origins of health and disease as well. So I would also welcome anyone who is interested in reading more or submitting a manuscript to the journal. And finally, I'll put an advertisement out for the World Congress, which is occurring in Vancouver, Canada, at the end of August. That means quite a bit of feedback. I am so sorry. Me. Okay, I'm going to carry on speaking because I'm just going to wrap it up before Professor Safri can take over for me. I would like to invite everyone to please join us in Canada and Vancouver if you're interested in maternal and child health, and certainly we'll have lots of different sessions discussing maternal and child health, as well as the role of agriculture in shaping many generations to come. So I'll wrap it up there, and thank you so much for giving me a couple of minutes and speaking on behalf of the International DOHAD Society. Thank you.
Thank you so much, Professor Sloboda, for your enlightening remarks. Now I invite one of the mentors of today's webinar, Professor Richard Safri. Yes, is it audible?
Yes, it is.
Okay, sir.
Well, thank you very much, Dr. Pooja, and thanks so much to Professor Sloboda, Dr. Pahil, Dr. Georges, Dr. Singh for your previous remarks. I too would like to say good morning, good afternoon and evening, depending on where you are. Namaste. On behalf of the international team, I'd especially like to thank Professor Bell for his ongoing passionate advocacy for research in India. He truly is an inspiration to us all, and for his invitation to join this important meeting on women and neonatal health. It's particularly relevant, as we all know, on International Women's Day. It's strange when we have to have a talk to tell us why women are important in agriculture. I think that says a lot about the current problems women face internationally. I'd like to pay my respects also to the patrons and the organising committee who made this meeting possible. This truly is a very important area of need, particularly in India, which unfortunately has some of the worst clinical outcomes internationally for women in terms of pregnancy and early childhood health. We've already heard that around 20% of all pregnancies in India end prematurely with a preterm birth, and that has a whole range of adverse flow-on health outcomes for the children. We haven't heard that 10 to 15% of pregnancies are complicated with gestational diabetes, another 10 to 15 with preeclampsia, both of which are also represent considerable burden not only to pregnant women but to their offspring, who are at higher risk of a whole range of conditions later in life. And then again, there's the stillbirth data, which I'll come to in a minute. So according to the 2015 data, India was ranked first in the rate of stillbirths and neonatal deaths internationally, with over 1.3 million in total, while it ranks second for maternal deaths during or after pregnancy, with 50,000 in total. In 2017, between 3 and 5% of all children born in India died before their first birthday. So they're very alarming statistics, not only for in totality, but you think of all the individuals that are impacted by those poor outcomes, most of which you can attribute to some sort of environmental exposure. Yes, genetics does play a role, but environment is equally, if not more, in fact, probably more important. I shall also try and share my one slide. Bear with me. I've not had much success with this today, but we'll see how we go. Oops. So as I mentioned, environment, hopefully you can see a slide now. Yes or no? There are a range of environments that women are exposed to in pregnancy, most of which humans did not evolve to deal with. And so not surprisingly, those in the human body and the human pregnancy is not evolved to deal with such stresses, and they include things such as alcohol and smoking exposure, Western diet, fatty foods, sugary foods, a sedentary lifestyle. Some of these are not really relevant, particularly to an Indian setting, but other things such as pollution, air quality, a whole range of environmental toxins, heavy metals, they're very, very relevant in India, and in total, they're predicted to account for several million deaths annually if you look at the Lancet recent series on the cost and burden of environmental exposures. So, of course, the key environmental factor that most people on this call are interested in is nutrition, and nutrition is incredibly important, and that is something that has always been important, including diet in pregnancy, but also early postnatal nutrition and breastfeeding. So all of these environmental exposures, they don't work in isolation, they work together, and that's why it's so hard to understand their individual roles and how they impact the health of a pregnancy and the early outcome. So the mediating mechanisms that are thought to convey this risk through development and early development are widespread in their nature, but generally they involve the gut microbiome, which you'll hear a little bit about today, epigenetic processes, which I study, which you won't hear much about today, and inflammation, which you'll also hear a little bit about today. In combination, these set up a molecular and cellular blueprint in a baby prior to birth that can be transmitted and have an impact throughout life. And there's a lot of data now, particularly in animal experiments, to support that effect. What's most concerning now is data, and if you look to the right, I don't know if you can see this, but there's now emerging evidence of intergenerational effects. So an exposure of a mother to an adverse environment can impact not only her developing fetus and therefore the baby she's carrying, but also that baby's subsequent children, because that baby in utero is carrying the germ cells that will make the next generation. And it's now very clear that the germ cells very, very early in development can be modified in a way that can transmit risk across generations. So it's very complicated, it's fascinating, but the important thing is that nearly all of these exposures are modifiable, but to modify them, you have to have clear evidence that they're having an impact. Otherwise, the public health message just gets more or less ignored. So today we're going to have 4 wonderful talks that will deal with some of these issues. We'll hear from Kate, who'll talk a little bit about the host microbiome relationships, early life and aging, and understand some of their role in modulating health and disease risk. Naraya will tell us about the importance of social isolation, and I didn't mention that stress is one of the most, let's say, toxic, for want of a better word, exposures you can experience in pregnancy. And we all know the stress and the psychological impacts that social isolation of our current lifestyle through this pandemic. It's very worrying to understand the risk that they may have in the future. She'll talk about that and the relationship to inflammation in a rat model, I believe. I'm not quite sure what Dr. Ruby will present, but I'm looking forward to it anyway. And finally, Toby from my group is going to tell us a little bit about the role of early life infection and inflammation and how this might set up a trajectory of risk for a range of later life cardiovascular events. So that's a mouthful. It's a lot to take in, but it's really only 1% of what I'd like to say. So I hope you enjoy this meeting, and I really sincerely hope that this leads to further discussions. As Deb so beautifully put it, that there's a huge DOHAD field out there who are waiting to engage with people such as yourself in this sort of research. So do please reach out to the DOHAD groups, and if you can get to Vancouver, it's a wonderful group of people. It's a fantastic meeting and it will inspire you, I'm sure. I hope we do get to meet again soon, and hopefully that's in person. So enjoy the meeting, enjoy your day or your night or your evening, and hope to see you soon. Thank you.
Thank you, Professor Richard.
Thank you, Professor Richard Seffrey, for your valuable remarks and enlightening us about the pregnancy in mother and health of children and adults. I now cordially invite Dr. Kate Kennedy to deliver her keynote lecture on host microbe relationships, impacts on the mother and offspring. Dr. Kate completed her PhD on the role of maternal gut microbiome in perinatal programming in the lab of Dr. Sloboda at McMaster University, Canada. She previously completed her BSc and MSc in biology at the University of Waterloo. Her research explores host microbiome relationships in early life and aging to understand their role in modulating health and disease risk. Dr. Kate Kennedy, please join.
Thank you, Dr. Malik, for that introduction. And thank you to all the organizers for inviting me to share some of my research today. I'm going to try, I think that's showing my slides now.
Yes, ma'am, visible.
Okay, thank you. So again, thank you for having me here today, I'm still in yesterday here in Canada. I'm still on Monday, late at night, and I'm happy to be here to talk about the maternal microbiome relationships in pregnancy and impacts on offspring intestinal development. So when we think about what determines whether we're healthy or not, what determines our health potential, most people, I think, would mention genetics, diet, activity as factors that contribute to disease risk. But something that a lot of people don't consider is the impact of early life exposures. And when we're looking at the impact of early life exposures, an important thing to keep in mind is that it's really an interaction between the early life exposures and the postnatal environment that contributes to either health or disease risk. So in the field of DOHAD, which has come up a few times now in the welcoming remarks, Dr. David Barker initially described this relationship between birth weight and risk of type 2 diabetes as an adult, where increasing birth weight from a lower end of a normal birth weight decreased the risk of type 2 diabetes as an adult. And then there's this slight increase again as you increase birth weight. So there's this J-shaped curve. And after this was initially described, a large number of studies have since come out looking at birth weight and correlating it with disease risk for a number of different diseases later in life. But it's important to keep in mind that birth weight itself isn't what's contributing to disease risk. It's just a marker of the prenatal environment. And low birth weight might be an indicator for a poor or inadequate early life environment. And as I mentioned just a second ago, it's also important to keep in mind that this increased risk of disease is due to a combination of that early life developmental environment and the postnatal environment. And a book written by Peter Gluckman and Mark Hansen described this combination of early life and postnatal life and the mismatch between those environments, where human beings now are increasingly mismatched to their environment. And this impact of this mismatch is what's driving an explosion in diabetes, heart disease, and obesity in adults. So one of the reasons behind this is the idea of a predictive adaptive response. So before a baby is born, it gets signals from the mother in the form of hormones or nutrients or oxygen, and it uses those signals to make a prediction about what the environment is going to be like that it will be born into, and it uses that information to adapt to that environment that it's predicting. But the predictions that it makes might not be accurate, and the adaptations might lead to an increased risk of disease if they are not appropriate adaptations. So in the research that I do, I am interested in figuring out how the fetus can be misinformed and have that mismatch with its postnatal environment. And specifically, one of the misinforming cues that I am interested in is the impact of maternal obesity or caloric excess, nutritional excess. So maternal obesity can impact maternal adaptations to pregnancy, and then in pathways similar to those predictive adaptive responses, this can impact fetal adaptations, and then that could be leading to increased risk of disease in the offspring later in life. So the metabolic adaptations are an important part of this during pregnancy. And in a normal pregnancy, there are a number of adaptations that the mother makes to her metabolism to support the development of the fetus, including things like decreased insulin sensitivity, increased circulating triglycerides, increased gastrointestinal transit time. And interestingly, a lot of these metabolic changes in pregnancy are mirrored in the metabolic changes that we see in non-pregnant obesity. Things like, again, decreased insulin sensitivity and increased circulating triglycerides. And something that for coming up on 20 years in the next few years has been recognized as a contributor to obesity is the gut microbiome, or the bacteria that live in our intestines. And one of the landmark studies that showed a causational role for the gut microbiome in obesity looked at twins who either were or were not obese and put their gut microbiome into germ-free mice and showed that the gut microbiota from the obese twin induced an obese phenotype in the mouse that it received that microbiome. And interestingly, a similar study was done in pregnancy by Corin et al. Back in 2012, where they took the gut microbiota from a woman in the first trimester of pregnancy or the third trimester of pregnancy, and again put it into a germ-free mouse, and showed that the third trimester microbiota induced a lot of those metabolic changes that are associated with the third trimester of pregnancy. So it seems like the maternal gut microbiota has an important role to play in maternal metabolic adaptations to pregnancy and also in maternal obesity potentially. So since that paper came out in 2012 first showing this relationship, a number of other studies have come out looking at different aspects of maternal metabolism during pregnancy and the gut microbiome, including looking at early pregnancy in women with hyperglycemia or normal blood glucose and connections with metabolic hormones and overweight and obesity, and gestational weight gain in the mothers and the gut microbiota of their infants. So just to summarize a few of the main findings of some of these studies, obesity is associated with a decreased alpha diversity of the gut microbiota, and that just means within an individual how diverse that microbial community is, so how many different species there are and how evenly distributed those species are, as well as an increase in inflammatory markers. And interestingly, obesity, excess gestational weight gain, and gestational diabetes are all associated with a decrease in bifidobacterium, which is an important bacterium for the infant gut microbiota. So although there have been studies looking at the microbiome during pregnancy and the relationship with maternal obesity and gestational weight gain, there's really been a lack of studies looking at the microbiome over the course of pregnancy. Most of these just look at one time point. So something that we are doing in collaboration with Dr. Braun of Berlin is a study in humans characterizing the gut microbiota over the course of pregnancy. So getting samples at first, second, and third trimester of pregnancy, as well as at delivery and at 6 months postpartum. And we want to investigate how maternal BMI and gestational weight gain impact the pregnancy-induced changes to the gut microbiome. And I'm currently working on writing this up, and hopefully we'll be submitting soon. And I hope that some of you might be interested in reading that when it's published. So it seems like the maternal gut microbiota has important roles in mediating maternal metabolic adaptations to pregnancy, and that by itself could lead to changes in fetal development and offspring disease risk. But the maternal gut microbiota also seed the infant gut microbiota. During delivery, the first bugs that the infant is exposed to are its mother's microbes. And this has been also shown by a number of studies looking at the transmission of specific microbes from mothers to their infants. But one question that recently in the past few years has arisen is, when does this relationship first happen? And is there potentially transfer from the mother to her baby of microbes before birth even happens? So back in 2014, this paper came out looking at or suggesting that there is a unique microbiome in the placenta. And a couple of years later, there was a rebuttal of this paper saying that the findings of the first paper showing a placental microbiome were actually due to contamination, and that inadequate controls were used in that study. And this really sparked the beginning of the debate between the sterile womb and in utero colonization. Are there microbes present in the fetus or placenta before birth? So similarly, back in 2016, it was first suggested that the human gut could be colonized before birth in fetal life. And we just last year published a study that I'm going to go over some of our data for, essentially saying that there is not colonization before birth, and some lessons learned from some of the studies that suggest that there is. So one of the biggest problems to overcome when we're studying something where microbes, if they are present, are at very low abundance is the problem of contamination and deciding what is an indicator of colonization versus what is potentially just contamination. So to work on solving this problem, in collaboration again with Dr. Torsten Braun of Berlin, we came up with a collection strategy to reduce contamination in a human cohort. So first problem in determining whether the fetus is colonized is if a baby is born vaginally, the baby is exposed to its mother's microbes during delivery. So all of the infants in our study were delivered by cesarean section. And furthermore, it was all primary cesarean to avoid any confounding factors for infectious causes of secondary cesarean section. And also, even during a cesarean section, as soon as the uterus is exposed to the external environment, there are microbes present. So to minimize contamination during delivery or prior to delivery, we only included breech deliveries as depicted in the little image above, where before the baby is even born, we're able to collect our samples. And then finally, there are microbes on maternal skin. So we had an extended aseptic area and modified drape to prevent contamination from maternal skin. So I will just briefly show one of the figures from our paper that I think really captures the main idea behind our findings. And I'll just briefly describe this. So this is a dot plot of genera that were present in the various samples, where each row is a sample and each column is a different genus. And then the rows are broken up by the type of sample that they are. So the very bottom box is showing our infant samples because we compared our meconium samples or our fetal stool samples to 6-month-old infants. And you can see that there are a lot of different genera present there. There's a lot of diversity in the infants. And then we also compared our fetal meconium samples that we collected to a neonatal meconium, which was first pass meconium collected after either cesarean or vaginal birth. And those samples are fairly heterogeneous, where some of them have a lot of diversity. Like this one that has points all, oh, I guess you can't see my pointer. This one that has a lot of different genera present in it, whereas some of them have very little bacteria. And then the top two boxes are our fetal meconium samples and our negative controls. And what we found was that pretty much all of the genera that we found in our fetal samples were also present in our negative controls. And it isn't shown in this figure, but some of them you can see show up in the fetal samples and not in the negative controls. But we also analyzed these samples across 2 different sequencing runs, and none of the samples or none of the genera that show up in the fetal samples were consistently showing up across both of those runs. So what these data show us are that, first off, robust controls are essential for low biomass samples. You cannot tell if something is contamination or a true signal if you don't have enough negative controls included. And then secondly, and importantly, these data show that we're not normally colonized before birth. So there are cases where bacteria are present in the case of infection, but in healthy pregnancies, the fetus is sterile. So these findings were really interesting to us because if the fetus is sterile before birth, this opens a window of opportunity to modulate the microbiome. And there are a number of different time points that we can modulate it at. So maternal factors can contribute to the infant microbiome through the transmission from the mother to the infant during and after birth. Neonatal factors like mode of delivery, gestational age have also been shown to modify the infant microbiome. And postnatal factors like feeding mode, breast milk or formula, host interactions, maternal diet, have all been linked to the infant microbiome. So the fact that we can know now when colonization first happens means that we can intervene to improve health throughout the life course. And just to bring this back to the big picture, the reason that this is so important is because the earlier that we're able to intervene in improving health and reducing disease risk, the bigger the effect we're able to have. So this is just a hypothetical disease risk over the life course. And it's showing that if we're able to improve health or reduce disease risk early on, towards the end of life, that has quite a large effect compared to an intervention later in life that won't have that same magnitude of a reduction in disease risk. So I just want to wrap up and say thank you to everyone for listening to my talk, and thank you again to the organizers for inviting me to speak. And I'd like to especially thank our collaborator, Dr. Torsten Braun, as well as Dr. Michael Surratt, who I work with on microbiome analyses, and a special thanks to my PhD supervisor who mentored me through all of these projects and continues to mentor me, Dr. Deborah Sloboda. Thank you for listening.
Thank you so much, Dr. Kate Kennedy, for your thought-provoking lecture. I now invite Ms. Naira Lopes for her keynote address on the effects of prenatal maternal stress due to social isolation on pregnancy and newborn outcomes, a model for COVID isolation stress. Naira Lopes is a research scholar investigating the generational impacts of maternal prenatal exposure to psychological stress in both mothers, fathers, and offsprings of rats. She is also investigating whether an enriched environment mitigates the effects of stress measured through uterine inflammatory and stress marker profiles. Ms. Naira Lopes, please join, ma'am.
Thank you for introducing me. Can you hear me well? I don't see you, but I hope you're listening to me. Let me confirm. Okay, I'll start my presentation. Today I will be talking about the effects of prenatal maternal stress due to social isolation on pregnancy and newborn outcomes, a model for COVID isolation stress. To understand the influence of the environment on maternal and infant outcomes, we need to know the concept of exposome. The concept was first introduced in 2005 by Christopher Wilde, and it gathers exposures a person can have over a lifetime from different angles. It is a complementation of the genome to understand disease risk in humans. It covers the external environmental exposures, such as climate changes and pollution. And it also includes personal factors like smoking, diet, exposure to
It's my humble request to all that kindly keep your mic off. Dear participants, kindly keep your mic off.
Thank you. Lastly, we'll take into account how these processes will affect internal responses to the body, like aging, epigenetics, gene expression, and so on. Understanding how these environmental exposures can affect our health is essential, and we can come up with ways of treating or mitigating this effect on our health. Fetal development is dependent on 4 major factors: the structural development, epigenetic factors, intergenerational and transgenerational epigenetic inheritance, and maternal and paternal factors. First, genetic factors provide a blueprint for fetal development, containing instructions needed for organism to grow, develop, survive, and reproduce. However, early life experience can shape organ structure and function, causing changes that persist over a lifetime. The brain is the most susceptible organ to environmental factors, and early life stress can affect brain plasticity, which is the ability of the brain to adapt to multiple stimuli, and cause changes in brain function and behavior. The second major factor associated with fetal development is epigenetics. This is the study of how the environment affects the way our genes work without affecting the DNA sequence. Some of the mechanisms that we know today are DNA methylation, histone modifications, and non-coding RNAs. The early developmental period is thought to be one of the most susceptible times for epigenetic insults. And this is because the DNA synthesis rate is high and the epigenetic modifications required for normal tissue development are established at that time. It was shown that insults during gestation can cause DNA methylation patterns and microRNA changes in the placenta, and that would increase the risk for pregnancy complications, such as preterm. Every tissue is exposed to genetic modification, but especially germ cells, that they can acquire epigenetic modifications that is transferred to the offspring, producing a phenomenon that we call intergenerational and transgenerational inheritance. That is the next factor that influences fetal development. To exemplify here, I'm using rats to demonstrate these effects. When a rat is exposed to any type of stress, its somatic cells go through epigenetic modifications. The direct effect of stress in the female lineage is different than the males. So first I will start with the female lineage. Here, when an F0 mother is exposed to stress, it's developing fetus, or F1. Yes.
We can't see your slides.
Oh. Let me try to start again. Can you hear me well?
We can hear you. We can't see your slides.
Let me try again. Sorry for that, everyone. Can you see it now?
Yep, that's good.
Yes, yes, yes.
Oh, sorry for that. Let me start again this slide. First, I will start with the female lineage, okay? When an F0 mother is exposed to stress, it's F1 generation, which is a developing fetus, and it's germ cells that will produce the F2 generation, that is the grandchild, are directly exposed to stress. Here, an intergenerational epigenetic inheritance represents the transmission of epigenetic marks from one generation to the other, with direct exposure to stress. The F0 generation, or the great-grandchild, is the first generation not directly exposed to stress and can be considered a truly transgenerational inheritance. However, the exposure of the F0 fathers can directly impact the sperm that will fertilize the oocytes, producing the F1 generation, and that it's an intergenerational transmission. The F1 will breed and produce the F2 generation that is the first unexposed generation in this lineage, presenting a truly transgenerational phenotype. The conclusion here in this slide is that stress experienced by our great-grandmother and great-grandfather can affect health outcomes across generations. Now, we will be talking about the effects of maternal prenatal stress on fetal development. On this figure, you can see that environmental factors like diet, pollutants, and also psychosocial stress can cause changes in the epigenome or genome, reproductive organs and cells, and that can affect multiple tissues, immune cells, and the microbiome. Stress exposure during these critical times of development, like the prenatal period in early life, can predispose an individual to diseases later in life, including the risk of preterm birth and other complications. We know that maternal stress has been associated with an inflammatory environment in the placenta and in the uterus, affecting the fetal environment and leading to poor infant health. In fact, the birth is an inflammatory event. In preparation to labor, uterine tissues express many inflammatory genes, and there is an infiltration of the uterus by leukocytes that produce pro-inflammatory cytokines and chemokines, as you can see here, to amplify the inflammatory cascade and amplify the inflammatory response. These signals stimulate the expression of proteins that finally induce myometrium contractions and other events leading to birth. Maternal stress, as you can see at the top, can induce early activation of the inflammatory cascade, causing preterm birth. Not only mothers, but also fathers play a big role on fetal development. Stressors, like we mentioned before, can affect the epigenome, sperm health and reproductive organs, and so on. These altered sperm can transmit marks of stress to the offspring, possibly via epigenetic mechanisms. However, it is important to know that indirect effects of paternal behavior can also influence child development, and these factors can control risk of diseases later in life. New evidence has shown that the seminal fluid also transmit signaling agents that interact with female reproductive tissues to facilitate conception and to modulate pregnancy outcomes, increasing the risk for preterm birth. Here are multiple examples of stress experienced by humans. Natural disasters, social disparities, poor access to care, chronic diseases, refugee status, all can be perceived as an intense stress affecting pregnancy and child outcomes. In particular, psychosocial stress has been shown to increase the risk of pregnancy complications such as preterm birth and also cause low birth weight. It also affects the central response to stress, leading to HPA axis dysfunction and cognitive changes. And these effects can extend from childhood through adolescence. A major stressor currently experienced around the world is the COVID-19 pandemic that has impacted every aspect of human life. To contain the spread of the virus, governments around the globe implemented an unprecedented worldwide social isolation for those exposed or infected with the virus, creating social isolation. Loneliness and social isolation are different concepts. Loneliness refers to the subjective feeling of being alone, while social isolation is a lack of social contacts and having few people to interact with regularly. Both terms are related to each other and can occur at the same time. Even before the pandemic, social isolation and loneliness were considered a behavioral problem across Europe, the United States, and China, and it only became worse with the imposed restrictions. Studies highlighted the detrimental impacts of social isolation and loneliness on various illnesses, including cardiovascular disorders, inflammatory, neuroendocrine, and cognitive disorders, and increases the risk of early mortality. However, little is known about their effects on pregnancy outcomes and child health. To address this gap, we subjected rats to social isolation before conception and during pregnancy, as you can see here depicted by the squares. The F1 offspring were split into two stress groups, the transgenerational or TG and multigenerational group or MG. Transgenerational refers to stress exposure in the parental F0 animals only, while in the multigenerational group, all generations were subjected to social isolation. The rats were individually housed until delivery, when they were kept with their pups until weaning of the offspring. We recorded gestational length and collected uterine horns at lactational day 21 to assess inflammatory markers involved with parturition. We showed that rats had shorter gestational length in the F0 generation only, as you can see here, and we did not see any changes in pregnancy duration in both transgenerational and multigenerational offspring. We also measured corticosterone hormone levels in plasma, which is a stress hormone, and we could see only increased levels in the F1 offspring of both groups, suggesting that rats are becoming more resilient to chronic social isolation. However, even though the corticosterone levels were only increased in F1 animals, we found reduced neonatal weight in F1 and F2 generations due to both transgenerational and multigenerational social isolation stress. And this finding was observed in both sexes. When we analyze pro-inflammatory cytokines in uterine tissues of rats, we show increase in interleukin-1 beta mRNA expression in the F0 generation, and a decreased expression in the F1 to F3 generation in the offspring. Interestingly, this finding was present in both stress paradigms and suggests that the offspring might be developing adaptation to the stress. In contrast, we found an upregulation of the interleukin-1 receptor 1 expression in animals experiencing cumulative effects of social isolation. And a previous study found that increased interleukin-1 R1 expression and protein abundance in rats is increased near parturition, which may suggest here in this study an inadequate adaptation. And supporting our previous findings, interleukin-1 beta abundance was significantly reduced in F1 uteri of the transgenerational group compared to controls. And also we saw a trend to decrease interleukin-1 alpha protein abundance in the multigenerational group. To conclude, we found that social isolation stress might affect the parental generation more than the offspring, illustrated by shorter gestation and increased interleukin-1 beta pro-inflammatory cytokine expression in the uteri of these rats. And we also suggest that offspring are becoming more resilient than their parents due to chronic social isolation. If we could translate these results to humans, it would indicate that social isolation stress might be a stress to parents, but the transmission of stress to the progeny would be much less present when compared to other stressors that we see in the literature. Finally, it is important to understand the biological consequences of social isolation stress to expand the awareness of the risks of lack of social interactions. And these findings might facilitate the development of strategies or interventions to mitigate the risk of adverse pregnancy and offspring outcomes due to stress. We end up with a debate, nature or nurture. What is the most important factor to influence our health outcomes? I would say it's probably a combination of both genetic code and environmental experiences. And we have to keep in mind that positive experiences promotes neuroplasticity and behavioral performance. We can mitigate the effects of stress by improving our environment with actions such as practice of physical activities, good nutrition, good parenting, music, and therapy. By maintaining a healthy lifestyle, we may reverse the effects of prenatal and ancestral stress. Thank you for listening, and I would like to thank our collaborators, Dr. Gerlinda Metz from the University of Lethbridge, and also my supervisor, Dr. Olsen from the University of Alberta, for guiding me throughout my PhD. And also, thank you for inviting me to present at this meeting.
Thank you so much, ma'am, for your illuminating lecture.
Thank you.
Now, Professor Ruby Bhatia, she is heading the Department of Gynecology at MMISR Mulana. She has served on various positions in different institutes in India and fellow of prestigious society engaged in promoting women health. She is vice president and life member of Society for Fetal Medicine. Safe motherhood and neonatals is close to her head and heart. In this context, I cordially invite her to deliver her keynote address on safe motherhood and neonatals health. Professor Ruby Bhatia, are you there, ma'am?
Yes, I am uploading. Good morning, all.
Okay, ma'am, good morning.
Good morning, all. Very warm greetings. First of all, I shall thank Dr. Bahel and Madam Pooja for making me part of this great international academic event, where we all are concerned about motherhood, the mother and the child health. I shall talk how safe motherhood can minimize maternal and fetal risk. I'll just upload my presentation. My presentation is visible?
Yes, ma'am, visible. Okay. It's visible.
UN Secretary General, the quoting is, to achieve the every woman, every child vision and the global strategy for women's, children and adolescent health, we need evidence-based approaches to antenatal care, which aims to put women at the center of the care, enhancing their experience of pregnancy and ensuring that babies have the best possible start of the life. Safe motherhood is the birthright of every woman. That's why we celebrate a Safe Motherhood Day every day. We must raise awareness about respectful healthcare to expectant and lactating mothers. There was a campaign in 2017 where over 1 lakh women across our country were asked that what would be their priority, what do they want? And the maximum number of the women replied that they need personal, reproductive, and maternal care. So we, in all developing countries, aim to provide respectful maternity care through various program charters, and Lakshya program remains one of them. In a campaign in 2019, Midwives for Women, it was planned that all midwives will be trained to do safe vaginal delivery. And we also run Janni Shishu Suraksha Karyakram in India so as to provide and promote institutional free deliveries with free package for everything for 1 week, 3 to 7 days after delivery. To the world, a woman is a mother, but to your family, she remains the world. Safe motherhood has 4 pillars which form the cornerstone to prevent maternal and fetal risks: family planning, quality antenatal care, clean and safe delivery, and basic essential obstetric care. Quality antenatal care is critical, and evidence-based actions related to health promotion, disease prevention, screening, and the treatment of the causes is mandatory. This reduces complications from pregnancy and childbirth, reduces preterm births, stillbirths, and perinatal deaths. Patients' education and health promotion is essential for pregnant women. She must be aware of the danger signs during pregnancy, like headache, visual disturbances, fainting or dizziness, and any onset of uterine contractions, cramp abdominal pain, decreased fetal activity, and must report to the skilled birth attendant and the healthcare facilities for the same. WHO says every pregnant woman and newborn receives quality care throughout pregnancy, childbirth, and postnatal period. This will go a long way in reducing the mortality and morbidity, both for the mother as well as for the fetus. Women should have a positive pregnancy experience from quality antenatal care, and this should be transitioned to a positive experience during labor and birth, followed by a positive motherhood with a maternal self-esteem, competence, and autonomy. WHO 2016 for antenatal mothers provides the proforma or the setup for the schedule of antenatal care. The first visit must be by 12 weeks, and the next should be at 20 to 26 weeks with total of 8 antenatal visits. And if she does not go into labor till 41 weeks, she must report to the healthcare facility for delivery. On the first very registration visit before 12 weeks, we need to assess for high-risk factors or the risk to the mother and the fetal health with a detailed history and examination, and the blood pressure recording remains mandatory as preeclampsia and eclampsia forms the deadly triad for maternal mortality. A hemoglobin checkup to rule out anemia, urine culture to rule out infections, and a glucose challenge test for any evidence of gestational diabetes mellitus, ABORH grouping of both partners, VDRL, HIV and HBs antigen, and thalassemia screening in high-risk endemic areas. The women must be counseled that she may have nausea, vomiting, craving and aversions, heightened sense of smell, mood swings in the first trimester, whereas in the later half, she may have abdominal pain, and breast changes due to pregnancy, abdominal stretch marks, and she will start feeling the baby. In the third or the later half of the pregnancy, she will have strong fetal movements as kicks. She can have swollen feet or abnormal edema over hands and face, and she may have leaking from the breast, or she may have frequency of micturition. After delivery, she may feel fatigue, she may have abnormal discharge, so she should be counseled about all the minor ailments or the risk factors in the pregnancy. She should be counseled to note for any high risk factors during the first registration visit if she is more than 30 years, or she is short stature, or there is any history of bleeding in the first or the later half of the pregnancy, any malpresentations or malpositions, more than one pregnant baby, that is twins, or excessive fluid in the abdomen, any history of rise of blood pressure in form of preeclampsia or eclampsia, anemia, prolonged pregnancy, previous history of a stillbirth, preterm birth, IUD, or previous intervention for manual removal of placenta, previous cesarean section, or a previous instrumental delivery, or pregnancy associated with any medical disorder like cardiac disease, diabetes mellitus. So it makes the pregnancy high risk, and she needs more frequent visit than the 8 visits prescribed by WHO. We must emphasize on the nutritional interventions that she must eat healthy to be physically active during pregnancy, with increasing energy and high protein diet, balanced protein dietary supplementations. Every pregnant woman in the developing countries is given 100 tablets of iron, 100 milligram elemental iron with 500 microgram of folic acid, and she should start it after 16 weeks of pregnancy, continue for the 100 tablets, a deworming with tablet albendazole 400 milligram anytime after 16 weeks of pregnancy, and in all developing countries, high dose calcium 1.5 to 2 gram should be supplemented to prevent the risk of preeclampsia. There is no role of nutritional interventions in the form of micronutrients, zinc supplementation, B6, vitamin E or C. So she should be advised that she should have adequate grains, adequate grams, and adequate green leafy vegetables, and say no to the bad foods during pregnancy. We must estimate the gestational weight gain, and for a normal weight women, the average weight gain should be 11.5 to 16 kilograms during pregnancy. She should maintain a good oral hygiene with a physical activity of around 30 minutes daily walk. She should avoid smoking, alcohol, and misuse of drugs. And yes, during this COVID-19 pandemic, she should be advised to have a frequent hand washing, wearing of mask with social distancing. The women must be immunized for tetanus and diphtheria toxoid with 2 doses 4 to 6 weeks apart, and COVID-19 vaccination is perfectly safe in pregnancy as well as lactation. Prenatal counseling is a must-do in all high-risk cases. She must be advised to rest for 8 hours at night time and 2 hours in the daytime with a proper lateral sleeping posture. She must be advised and must be asked for any intimate partner violence. 1 in 3 women worldwide, they experience physical or sexual violence, mostly by an intimate partner. We should be very careful about it that hit, hurt, or pushed not only is not the violence, but any threat or made to feel afraid by your partner, or forced to have sex or do something you don't want to do, or kept away from your family, friends, or being controlled, they are also part of intimate partner violence. This domestic violence may have effect both on the maternal and the fetal health. There is high risk of abortions, preterm births, stillbirths, and also getting a dangerous vaginal infection in case of forced unprotected sex. There is increased chances of first and second trimester bleeding, and in many scenarios, they may lead to injury to the uterus also. The babies born to mothers with intimate partner violence can be low birth weight, preterm, they can be trouble in nursing or taking a bottle, may have sleeping problems, and may have harder to comfort than other babies. WHO recommends that hemoglobin must be above 10 or 11 gram in every antenatal woman at the first registration visit, and she must be tested for asymptomatic bacilluria and must be inquired for intimate partner violence. We must test the sugars by a glucose challenge test at the first registration visit and then at 24 to 28 weeks to rule out gestational diabetes mellitus. Use of tobacco should be stopped, smoking should be prevented, and drug abuse and alcohol should be avoided. In all antenatal women in developing country, at the time of registration, HIV testing, syphilis, VDRL or RPR must be done at the first visit and must be repeated in the third trimester in high-risk women. The fetal well-being should be assessed with daily fetal movement count from 30 weeks or 28 to 30 weeks onwards, maintaining a gravidogram by symphyzofundal height, and we should do a non-stress test in the later half of the pregnancy. A dating scan for NT-NB, that is nasal bone and nuchal translucency, must be done between 11 weeks, 3 days to 13 plus 6 weeks, and a targeted level 2 ultrasound to rule out gross congenital fetal malformations between 18 to 20 weeks, and ultrasound for fetal well-being must be done in the third trimester. So all women with recurrent urinary tract infection or asymptomatic bacilluria must be treated with antibiotics. Rh negative women must be given an antenatal prophylactic dose of anti-D immunoglobulins, and TD vaccine 2 doses should be given to all. In the endemic areas for malaria, the intermittent preventive treatment with sulfadoxine and pyrimethamine is recommended. Then oral pre-exposure prophylaxis with tenofovir disoproxil fumarate should be offered in the areas where there is substantial risk of HIV infection to the mother. Never, never allow any high-risk pregnancy to become post-date, to go beyond expected date of delivery. During labor, every mother must be giving respectful maternity care. A birth companion must be allowed during birth in all developing countries. This helps in spontaneous vaginal birth and reduces the incidence of need of analgesia, augmentation, or cesarean section. Now, United Nations SDG goal 2030 aims to bring maternal mortality ratio to less than 70 per 100,000 and safe motherhood remains a cornerstone for reduction of maternal mortality. So as to reduce the maternal mortality ratio, we have to give adequate family planning services, skilled birth attendant at every birth, emergency obstetric care, ultrasound for safe motherhood, and formulate various policies and infrastructure to avoid delay in seeking care, reaching care, and receiving care. The family planning services in developing countries, every girl, every adolescent girl, and every pregnant woman and postpartum women should have access to family planning, maternal health care, and the right to choose when and how often to have children. It should be the right of the women. We should give family planning services to avoid unwanted birth, for adequate spacing, and to delay pregnancy in adolescent girls. For every birth, presence of skilled birth attendant should be mandatory, and they should be trained for the same. All skilled birth attendants must be trained for emergency obstetric care in form of administration of antibiotics, uterotonic drugs, to deal with eclampsia, and administration of magnesium sulfate for manual removal of placenta, and perform instrumental vaginal delivery as well as neonatal resuscitation. All skilled birth attendants must be trained for eclampsia management, and we should try to do periodic measurement of blood pressure on every antenatal visit, so for the early detection of preeclampsia, and giving tablet Ecosprin 150 milligram once daily in all high-risk cases. All should be trained for intrapartum administration of magnesium sulfate in cases of imminent and eclampsia, administration of antihypertensive therapy, and all cases with preeclampsia should be delivered at 37 weeks of pregnancy. The postpartum hemorrhage management should be taught to all skilled birth attendant, and to prevent this, we should do active management of third stage of labor. Simulations and drills for management of postpartum hemorrhage should be taught, sensitized to healthcare providers, and they should be trained in using the drugs oxytocin, misoprostol, carbetocin, and tranexamic acid for control of postpartum hemorrhage. WHO also recommends the 4 cornerstones for safe motherhood. Ultrasound plays a great role for safe motherhood. It helps in the first ultrasound done between 11 to plus 3 weeks to 13 plus 6 weeks, helps in diagnosing extrauterine pregnancy, abnormal placentation or placenta previa, and early identification of molar pregnancy. It tells us about the fetal age, any risk of chromosomal defect by first trimester aneuploidy screen, and it also checks the neural tube defects as early before 12 weeks. Then a targeted anomaly scan must be done between 18 to 20 weeks to rule out major birth defects which are not compatible with life. A third trimester ultrasound for fetal well-being must be done at 32 weeks for ruling out fetal growth restriction or small for date babies. Doppler has a great role to play in safe motherhood. This can be practiced in fetal growth restriction, in fetal anemia in Rh negative pregnancy, in multifetal pregnancies, and in various placental cord and abnormalities. So we should warn every pregnant woman about the risk factors so that she can avail the healthcare facility, and she should have a contact number of the healthcare provider. Respectful maternity care in the form of Every woman has a right for information and privacy. She has right to choose and have a companion during childbirth. She has right to food and drink during normal labor, and she has right to liberty to move around during labor with audiovisual privacy, and she has right to choose the position of choice during childbirth. And we always make it that mother and baby should be kept together as far as possible. All women deserve respectful maternity care. So for this, we have to honestly practice respectful maternity care in our labor rooms, and so that this will increase the spontaneous vaginal births with minimum of augmentation of labor should be there. There should not be any verbal or physical abuse. We should not do immediate cord clamping, and there should not be any out-of-pocket expenditure for drugs, diagnostics, or for gracious payment to the staff for celebration of baby's birth. We must practice the 3 simple steps for neonatal resuscitation. Always dry and cover the baby, clearing the airways by suction, and inflating the lungs with air using ambu bag and mask where needed. Only remember that only 3% of the babies need resuscitation. And everyone attending the birth should know how to resuscitate. Every ANM, GNM, doctor, obstetrician. Do not wait that there is no pediatrician, so no neonatal resuscitation. Anybody who is doing a childbirth should be skilled enough for neonatal resuscitation whenever, as and when the need be. So essential physical resources must be available with the competent staff, and the healthcare should always be patient-centered. We in India are running various programs for safe motherhood so as to minimize the risk of maternal and fetal risks. We in our country are running Pradhan Mantri Surakshit Matritva Abhiyan, under which we are providing safe, free delivery with free transport, institutional delivery for every mother. Anemia Mukt Bharat, with providing of iron from adolescent girl till the birth of the baby to every mother. And we also run a Janni Shishu Suraksha Yojana, and we have various reproductive and child health programs under NRHM, and Lakshya, a labor room quality improvement initiative, is being run in our country in every district and tertiary care hospital. So I would end that a pregnant woman has got everything to take the world to her stride with the wings of safe motherhood. Thank you. Thank you, Dr. Bahel. Thank you, Dr. Professor Richard, Professor David, Professor Deborah, for giving me this opportunity to talk on safe motherhood. Thank you all.
Thank you, Professor Bhatia, for very educating lecture. Thank you, ma'am. Now I cordially invite Dr. Toby Mensah to deliver his keynote on the topic childhood infection, inflammation, and metabolomic and lipidomic profiles. Dr. Toby Mensah is a postdoctoral researcher at the Murdoch Children's Research Institute and honorary fellow at the University of Melbourne, Australia. His research is on the molecular processes underpinning the links between early life exposures, inflammation, and cardiometabolic health, and is particularly interested in the integration of multi-omic data. Dr. Mensah, please join.
Thank you for that, and thank you for the privilege of presenting my work here today. I'm just going to share my slides. Can you see those?
Yeah.
Great, thank you very much. So, as mentioned, I'm going to be presenting on work we've done investigating childhood infection, inflammation, and metabolic profiles. I'm not going to be talking much about the lipidomic profile today, just in the interest of time. But I'll give you a link to the manuscript where we discuss this in more detail. So first, I'd like to begin by acknowledging that the work I'm presenting today has had many different co-authors contributing to it, particularly under the supervision of Professor Richard Saffery and Professor David Bergner. I'd like to begin by talking briefly about cardiovascular disease, atherosclerosis, and how childhood infections might relate to this. So cardiovascular diseases, such as coronary heart disease and stroke, are the leading causes of mortality worldwide, with approximately 30% of deaths worldwide believed to be due to cardiovascular disease. These are some statistics of the burden of cardiovascular disease here in Australia. One Australian dying on average every 12 minutes from cardiovascular disease, and approximately 7.6 billion Australian dollars, which is 429 billion Indian rupees approximately, being spent on cardiovascular disease healthcare costs each year. And so cardiovascular disease is a large concern worldwide. One of the major contributors to the risk of cardiovascular disease is atherosclerosis, which is a thickening of arteries due to a plaque buildup. And there's now considerable evidence that atherosclerosis occurs across the lifespan, with even in newborns, some evidence of the thickening of arteries, indicative of the earliest stages of atherosclerosis. And so there's growing evidence that this process of atherosclerosis occurs across the lifespan, with arteries getting thicker, until it manifests as clinical cardiovascular disease later in life. And so there's many different health and lifestyle factors associated with atherosclerosis, but central to many of them is inflammation, and inflammation is believed to be a key driver in atherosclerosis. So with the evidence, growing body of evidence indicating that atherosclerosis begins from very early in life, we believe that inflammation exposures that an individual experiences across the life course from a very early age or even before birth, this cumulative inflammation contributes to this buildup of atherosclerosis over life. And so instead of thinking of cardiovascular disease risk as something that needs to be acted on in adulthood, we should be really taking this life course approach to cardiovascular disease risk. For example, there's now a lot of epidemiological evidence suggesting that severe inflammatory exposures early in life are associated with increased risk of cardiovascular disease. For example, infections in childhood are associated with increased risk of subsequent cardiovascular disease. And what might be occurring is that in these earlier stages of life, an increased inflammation burden accelerates this process of atherosclerosis, leading to earlier manifestation of cardiovascular disease. But that suggests that conversely, if we're able to reduce the burden of inflammation in early life, this might slow the process of atherosclerosis, resulting in lower cardiovascular vascular disease risk later in life. And so it's really these earlier stages of life where it's an ideal but overlooked opportunity to intervention to reduce cardiovascular disease before clinical symptoms start to manifest, with the aim of slowing this down so we are preventing the disease rather than trying to treat the symptoms afterwards. And so one of the inflammatory exposures that we're interested in is infection. So infections are a key inflammatory event, particularly early in life. And as mentioned, there's a growing body of evidence that childhood infections are associated with increased risk of cardiovascular disease later in life. And while inflammation is likely to be central to this relationship, the biological processes involved in the relationship between infections, inflammation, and cardiovascular disease risk are poorly understood. One process that might be involved is a relationship between infections and adverse metabolic profiles. So infection is associated to metabolic differences that are considered pro-atherogenic, in that these metabolic differences are associated with increased atherosclerosis and increased risk of disease in adults. So classic examples of these are higher triglycerides, lower high-density lipoprotein, which is often considered a good cholesterol, and lower apolipoprotein A1. However, while there's evidence in older children and adults that these infections are associated with these pro-atherogenic metabolic changes, infection burden falls disproportionately in young children, with infection being most common in sort of the early years of infancy. And there's currently no data on how metabolic or lipidomic effects of early childhood infection, what effects there might be of infection on metabolism at this early stage of life. So from infants in a population-based cohort in Australia, the Barwon infant study, we investigated the relationship between the number of parent-reported infections from birth to 12 months of age with plasma metabolomic and lipidomic profiles at 12 months of age. We also investigated whether markers of inflammation at 12 months of age were associated with differences in metabolic profiles. And lastly, we investigated the evidence for inflammation mediating a relationship between infections and differences in metabolic profiles at 12 months of age. So the Barwon infant study, or BIS, is a population-based pre-birth longitudinal cohort based on the Barwon region of Victoria, marked there on the map, just to the west of Melbourne. And this cohort is of just over 1,000 mothers and infants. Of these infants, we had 555 infants with complete infection data from birth to 12 months of age, reported at multiple time points. And we also, as part of BIS, have a lot of questionnaire and medical record data. So we were able to adjust for things like pregnancy exposures, such as gestational diabetes and pre-pregnancy BMI, and household income as a measure of socioeconomic position. And we were also able to adjust for birth outcomes, such as birth weight and gestational age, as well as early postnatal exposures, such as breastfeeding. And so I'm going to be talking today primarily about the metabolomic profile. So this is measured using nuclear magnetic resonance. Briefly, we've collected 12-month plasma from the infants, and we use a nuclear magnetic resonance to measure the metabolites in the plasma. So we get a spectra using NMR, and then from this we can quantify up to 228 different metabolic measures. So this includes glycate, which is a measure of inflammation I'm about to talk a bit more about, as well as amino acids, fatty acids, cholesterols, triglycerides, and other metabolites that have been implicated in cardiovascular disease and other complex diseases. Because 228 metabolic measures are a lot, I'm going to be focusing in on 51 measures that provide the most information in this data set. And so the advantages of using NMR to measure metabolomics is that this platform measures a range of metabolites that are relevant to clinical disease risk. And it's relatively low cost and high throughput compared to other methods, and there's also no batch effects. However, compared to other metabolomic platforms, such as those using mass spectrometry, it is less sensitive and it requires larger volumes of sample. And so the data I'm presenting today is from the Nightingale Health NMR metabolomic platform, and this has been used before in older children and adults, and there's been links reported between metabolomic measures on this platform and risk of diabetes and cardiovascular disease and all-cause mortality in adults. So that's the metabolomics data. The other data that this study was involved was quantifying inflammation. So inflammation is quite complex, and there's many different ways of measuring it molecularly. And so for the Barwon infant study, we wanted to consider how best to measure it cheaply and accurately. And so we used two different inflammation measures for the study. The first one is C-Reactive Protein, or CRP. This is an acute phase reactant produced by the liver. So when there's an infection or other form of inflammation, it's rapidly produced and increases dramatically over a couple of days and then returns soon to baseline. It has been used as a measure of chronic inflammation in adults, but it doesn't perform very well as a measure of chronic inflammation. There's some evidence that it can predict cardiovascular disease, and it's used clinically in risk stratification, but there's growing evidence that while CRP can be used to predict risk of cardiovascular disease, it's not involved in the process. It's just a biomarker. It's not mechanistically involved with cardiovascular disease risk. The other inflammation marker I'm going to be talking about today is glycoprotein acetyls, or glycate. This is a relatively novel inflammation marker, recently described, and it's a composite measure from NMR metabolomics. And it's believed that glyca is a measure of cumulative inflammation. So instead of CRP, which acutely increases and then decreases over several days, there's evidence that glyca can remain elevated for many years after a severe infection or inflammatory event. And there's now evidence from several adult studies. So on the right, there is a meta-analysis across many adult studies showing that glyca is a better predictor of cardiovascular disease and mortality than CRP. And so on the right, it is your odds of cardiovascular disease if you're in the highest quartile of glyca compared to if you're in the lowest quartile. And there's evidence that glyca and CRP are also giving different information about risk, and they can potentially both tell us different things about cardiovascular disease risk. So in this study, we measured high sensitivity CRP with ELISA, and which is an assay based on immunology biochemistry, and we measured glyca on the nuclear magnetic resonance platform. And just very briefly, it's probably not of interest to most people in the audience, but for all the measures, all the work I'm presenting today, all the metabolic measures have been log transformed. And we used linear regression modeling to investigate the association between each of the exposures, which was parent-reported infections or inflammation, and the metabolic measures as the outcome. And all the p-values here have been adjusted for multiple comparisons. And I'll also briefly discuss some mediation analysis afterwards. So this is a forest plot showing the relationship between the number of infections each infant had from birth to 12 months of age and their different metabolic measures at 12 months of age. So for each metabolite listed there, the estimated effect, if it's to the right of 0, it's higher with the number of infections you have, and if it's to the left of 0, it's lower with the number of infections you have. So the number of infections an infant had from birth to 12 months of age was associated with differences in several of the NMR metabolomic measures. And so I've highlighted those in red are metabolites that are associated with increased risk of cardiovascular disease in adults, and those highlighted in blue are associated with decreased risk in adults. And so you can see that with the more infections an infant has, the higher levels of these increased risk metabolites, and there's lower levels of these protective or decreased risk metabolites. So it looks like with the more infections an infant has, the more the metabolomic profile resembles those in high-risk adults. We were interested in investigating if these differences we see are due to infection causing inflammation, or if potentially they're acting through some other pathway. And so these forest plots, which are up now, are with number of infections as the exposure. But if we look instead at inflammation as the exposure, glyca, which is our measure of cumulative inflammation, we see that higher glyca is also associated with many of the NMR metabolomic measures. And this is in a similar pattern to the metabolic differences associated with infections, but more pronounced. We see stronger effects of higher inflammation than we do for higher infection. And so this is just bringing the two up side by side. And you can see that generally the metabolites that are higher with those in infants that have more infections, they're also the ones that are higher in those that have higher glyca. Just very briefly, we also considered hsCRP, which is our measure of acute inflammation. And while high hsCRP was associated with metabolic differences, these were quite a different pattern of metabolic differences to what we saw with higher infections and higher glyca. So it looks like that inflammation measured by hsCRP has a different kind of a relationship with metabolic health at 12 months of age. And that's just bringing up the two forest plots so you can see that they're quite different patterns of differences. And generally, we saw that infection, there's a strong correlation between the effects of infection and the effects of glyca on metabolomics, as shown in the scatter plot in the top right there. while there's a more modest correlation between the effects of infection and the effects of hsCRP on metabolomic profile. And so just briefly, we wanted to investigate the evidence for GlyK mediating the relationship between infection and metabolic differences. So how much of the relationship between infections and metabolic differences was due to increased GlyK. And it depended on the metabolite, but GlyK was estimated to mediate a substantial proportion, sort of between 23 and 78% of the relationship, particularly for triglycerides and high-density lipoprotein cholesterol. In contrast, hsCRP was estimated to mediate a much lower amount of the relationship, between 4 and 21%, which is similar to, so this suggests that, as we saw earlier, that GlyK is relevant to this relationship between infections and metabolic differences, while hsCRP is likely working on a different kind of pathway. So to conclude, we found that high number of infections was associated with metabolic differences that are associated with higher risk of cardiovascular disease in adults. And this is a similar kind of relationship as we saw with higher GlyK, which is a measure of cumulative inflammation. And the relationship between hsCRP and metabolites was distinctly different. And based on our work, we found evidence that suggests that GlyK, with higher inflammation, mediates the relationship between infections and metabolic profiles. So this suggests that early life infections may be contributing to the development of cardiovascular risk through inflammatory and metabolic differences that promote atherosclerosis. And that this relationship appears to be driven, at least in part, by cumulative inflammation as captured by GlyK. And this work is consistent with the work reported previously of relationships between severe infection in childhood and increased risk of cardiovascular disease in adulthood. And we've published, or we're in the process of publishing this manuscript, and the preprint for this manuscript is available on bioRxiv. And so this work here is investigating the relationship between childhood infections and inflammation and metabolites. But this is very relevant for considering pregnancy and neonatal health, as there's a lot of potential influences on how many infections a child has. So there's evidence that suggests that pregnancy exposures, such as gestational diabetes or exposure to maternal smoking, might be associated with childhood infection, as well as household income or other measures of socioeconomic position. There's also evidence that birth outcomes, such as gestational age, birth weight, and mode of delivery, might influence the number of infections the child has in early life, as well as breastfeeding in early life. So this work suggests that these early life exposures might contribute to the ongoing risk of cardiovascular disease across the life course through acting through inflammation. And with that, I'd like to thank you all for your attention and again for the opportunity to present and acknowledge everyone who contributed to this work. Thank you very much.
Thank you, Dr. Mensah, for your valuable lecture for research scholars and practicing doctors. I now request to Professor Rishi Bahel, webinar chair, to sum up the webinar, and there is a open session for the participants to ask their questions. Professor Bahel, are you there? Yes, is it audible?
Yes.
Now there is an open session if anybody wants to
Yes,
ask
yes,
any
yes,
question.
yes. Okay, I am here now. Yeah. I am happy to say that Professor David Olson has made the International Women's Day a really informative day for all of us, and selected very nice speakers.
Sir, kindly keep your camera on, Professor Bahel. Kindly keep your camera on
Hello. Yeah.
so we can see you.
Camera on. Hello. So, hello. So,
Yes.
the choice of speakers is very good and they have been very focused. And the sum up is that total effects from pregnancy to safe motherhood to what can happen if the early life infections are there, they are reflected into cardiovascular disease or diabetes at later stage. Kate Kennedy presented a wonderful lecture on the influence of microbiome, and Naira Lopes very elegantly demonstrated that the social isolations, the psychological isolations, not only impact the first generation, but transgenerations also. And her work tells every politicians that they should have rehabilitation program for those children who are born in COVID periods and suffering from one or the other problems, because if they are not settled right in this generation, they may affect the next generation. That is the indication of her research work, wonderful message she has given. Then Professor Ruby Bhatia has shared her long experience on how to ensure safe motherhood and how to protect the neonatals from various diseases. And she gave the package which is recommended by WHO and her own experiences, and mandated that there has to be a skilled birth caretaker. But in a country like India, where the economic disparity is too much, this remains a big guess rather than a reality. And we need to spread the education so that every pregnant mother knows herself that what is to be done. Okay, because of the economic reasons, it may not be feasible to engage a skilled birth caretaker. Okay? And more mainly in villages, this neo-rich people can afford it, and they are doing it also. But then the policies are on the papers, but they are not in the reality. Therefore, I suggest that the educational program should be modulated in such a way that there is a special teaching courses for the pregnant woman. The 8 registrations she suggested, it is very less known to people. Yeah? Many deaths occur because of this ignorance, and deaths of pregnant women and the neonatals. And in India, the death rate is alarming, as suggested by Professor Richard Savage, that many children below their first birth, before their first birth die. And this is alarming. This is alarming. As in SDGs 2030, as suggested by Professor Ruby Bhatia, aims at lowering this number very low. Okay, and again, Toby Mensah's description of his research work very elegantly lines up what happens during pregnancy, and if the infections are there in the fetus or in the early childhood, they may cause diabetes and cardiovascular diseases in the adults. And therefore, this lineup is very important, that we need to care for safe motherhood so that the chances are there. He has indicated that 228 metabolites can be identified, out of which 52 he has stressed. And one very good metabolite he suggested is high-density lipids and glyca, that be very useful in predicting or the adulthood diseases or in relation to infections, in early life. So these may be utilized by the practical doctors in a very good way. So in total, I really thank all the three mentors, to Professor David Olson, who conceptualized the whole webinar, Professor Richard Sepri, who extended it further, and Professor Deborah Sloboda, who also guided her students to present the research work done in our lab. So that is how I captioned them as the inspirators, supervisors, and moderators, because I was thinking what role I can assign to them. But they have very elegantly done their role in supervising the webinars, the inspiring the young ones. And this is real gain of International Women's Day. And the webinar dedicated to International Women's Day, I will suggest that 50 words or 100 words from each speaker, we can put it in some good journal as a summary of the webinar presented here. And if Professor Richard Safri, David Olson, and Sloboda, they deem it fit, then we can arrange that. This can be submitted to the Association of Indian Universities. Proceedings they publish every year. So one-page note we can always send it. So therefore, I will request every speaker to send me 50 to 100 words so that we can really document this webinar in much larger scope and in much larger benefit of the readers. So I thank once again everyone, and I hope that we will continue to get your cooperation in future also. We are planning to organize an offline conference, and I think that all of you will be helping us. Thank you very much.
Thank you so much, sir. This webinar's credit goes to you. It is next to impossible without your motivation, guidance, and support. Yes, sir. Professor DP Singh wants to communicate. Yes, sir.
For nice presentation. In Asia and then in Western world. Particularly, I was going through the newspaper. So, the area of women is one of the important for health, particularly
क्या हो रहा है पूजा?
very important issue.
Professor DP Singh, sir, your voice is not clear. Echo is coming.
Too much echo. So please check your mic. Too much echo.
Again, same problem, sir.
और दूसरे स्टूडेंट्स का तो नहीं है कहीं.
No, sir, it's not okay. Echo is coming.
अरे वो
Director sir, you kindly keep your mic off. Director sir.
Okay? Is it okay now?
Yes, sir. Now okay. Yes, sir.
So I was just saying, I thank all the speakers and organizers for organizing very nice seminars, and they gave lot of very important inputs on Women's Day. But the conditions in India, Africa are quite different from the Western world. Here,
Yes.
the drudgery of women, particularly in agriculture operations and post-harvest technology, is one of the most important issue which is affecting the health of women. Then another important issue is the empowerment of women. And for that purpose, as just narrated by Professor Well, education and skill development is also very important for the health of women and the children. I was just reading the newspaper in Bihar, which is very high populated state. One family has got 7 sisters and 1 brother. So the family parents, they decided that first education, then the empowerment. So every girl from that family is either in army or in post police or other operations. So the women which are empowerment, they are doing much good for health and also for their own issue. India has also launched a big program for the empower of women, particularly in the village panchayat or so, and also the reservation of women in every job. So that is another important issue. In Western, the cleanliness, particularly during the MC period, is also one of the a very important issue for Asia and Africa. Then in Western, particularly world, the overfed and obesity is another issue, while in Asia and Africa, the underfed is malnutrition is other issue. So I don't have to say much thing because already several things have been said, told, particularly in medicine and health and other things. Thank you very much.
So, Professor Richard, some sum up remarks from your side?
Sure, Rishi. Those talks were fantastic. It was nice to see different approaches to understand the pathways to poor health in maternal women and their babies, and to also get a better picture of the pregnancy landscape in India, which is quite a lot more detailed than I anticipated. There's a lot more testing recommended by WHO than actually happens in Australia. So I think if that is implemented, that's an incredible achievement in India, and that will solve a lot of problems. What I think is missing is a broader overview of how it all fits together. And so I like the idea of putting it all together in some sort of small written piece or written article for dissemination. I think David would help write that, and I'm happy to help with that as well. My question for you is, how do we turn this into some sort of research on the ground in India? I think that would be fabulous.
Yes. Yeah, you see that I did not hear before you people came about the preterm birth, and there was no conference also. So I suggested some of the people to file a public interest litigation with Government of India that we need to have a rehabilitation program for the preterm born, and we have to initiate programs to minimize the risk of preterm born by improving the pregnancy environment. And one lawyer from Agra has already done that on my saying. We can initiate research work, particularly at MMDU Mulana. This university has a wonderful multi-specialty hospital. It has a medical college. It has MD program, MBBS programs, and it has a research wing now. I am a part of research and development wing, and it is well-established research wing, so that we can initiate some steps to encourage people, youngsters, to come forward to apply for research grants, international as well as national grant. And you people can help us in designing the experiments so that we make a faster move. Even if we want to design experiments, we will take a while for becoming that wise to start meaningful experiment. We will learn by practice, but at the same time, if few people are around, maybe that we can establish collaborations.
Absolutely, but I think also Dr. Singh had made a very good point that education is also incredibly important, but also Dr. Bhatia and talking about the clinical care. So you should, I mean, bringing a clinical team plus an education team plus a research team together would be very powerful. So
we are likely to recruit, our director is likely to recruit a biochemist who has been working in the hospital, and she will be basically focused on the nutritional aspects for pregnant women. That is how we have selected her.
That's great. Yeah, I think there's a lot of potential there. That's a big problem to be solved. But it sounds like there's a lot happening already, which is fabulous.
Yeah, in India, this kind of research is very less. I'm telling you that it was 2018 when I learned the term preterm. And of course, the medical doctors, they know since last 10, 20 years, when the problem became very apparent, 23% of preterm born are in India. Yeah, we are highest in terms of preterm born children. And but in the academia, people did not know. Like me, I am considered to be one of the most aware person, but I did not know, frankly, honestly speaking. So this is, and when I go deep into thinking for this problem, it's really problem. It's not a problem of one generation. It is a problem of many generations. I learned that a person, preterm born child, has lower brain power, lower alertness, lower IQ. And if these problems, they are passed on generation to generation, then
there's a large economic
impact, not for this, for our generation. So that is why I think that we can emphasize this area of research. And I have identified 3-4 medical colleges, and I am talking to WHO on this problem, and they have suggested to form a foundation. I've told them the opera is already there, but they say that we can have a nutrition based foundation for starting this kind of work. The talks are in progress, and as soon as the MOA is signed, I will inform you.
That's a great idea. There are already some very good programs around the world, particularly in Africa. So there's a lot that could be learnt once you do set up a foundation, a lot of lessons that could be applied. It'd be interesting to get David's opinion, because he has a much clearer view of what's happening internationally. But I'm afraid I have to leave, but it was wonderful to see you all, and thank you.
Wait for 2-3 more minutes, because we have to perform a pleasant ceremony of vote of thanks.
Okay,
my
I've got
young colleague Jagdeep is there. Yeah.
So before that, I have a query from Dr. Bhatia. Ma'am, are you there?
Hello,
Yes,
I'm a JR working under Professor Dr. Ruby ma'am. I'm sorry to inform that she had to leave the conference because of an emergency call from OT ma'am.
Okay, but I have a query. As I am a mother of two, I want to ask a question that is painless delivery, that is epidural, is safe for the mothers, pregnant mothers?
Yes ma'am, it is being practiced, and in this era of advancement, we have a provision that some women who want to go for such procedures, then it is easily available. And other women, because other women who like there are some women who want to go for the normal delivery. For that also there are provisions. But obviously, the pain and all these conditions with the new advancement, this has been provided in a hospital, and it's easily can be taken care of. So I would like to say yes, it can be given and it could be advised.
Okay, ma'am. Thank you so much, ma'am.
Thank you, ma'am.
Now I request to Dr. Jagdeep Singh to propose vote of thanks.
Good afternoon all. Thank you, Dr. Pooja Malik, for assigning me a pleasant duty of proposing vote of thanks in behalf of organizers. I am Dr. Jagdeep, Department of Agriculture, MMDU, Mulana. It's immense pleasure to me to propose heartily vote of thanks to the Chancellor, Dr. Tarseem Gargji, and Vice Chancellor, Professor Harish Sharma, and other dignitaries in organizing committee to allow me and enable to us hold this prestigious international webinar on very precious International Women's Day, that is today, 8th March. The real mentor of today's international webinar, Professor David Olson, Alberta University, Canada, with own inspirational mentorship, persuasions, continued effort, and leadership of this webinar. As seen the light of the day, we sincerely trust that he will continue to guide and support us. I am thankful to Professor D.P. Singh, former Vice Chancellor, JNKVB, and President of SSARM, for his gracious present with us. Thanks to him for his valuable marks highlighting importance of nutrition. Our thanks are due to Dr. K.W. Georges, President of IFDDA, Germany, for his welcome greetings. His associations with our webinar is a source of encouragement. I am wholeheartedly thankful to Professor V.S. Pahil, Director and Dean, Department of Agriculture, MMDU Mulana, for his welcome addresses and highlighting on the role of women in agriculture. I am thankful to world acclaimed epigenetics Professor Richard Safari from Australia for his opening remark and setting the stage for the name of presentation by our speakers with his inspirational mentorship and supervision. I am thankful to Professor Debra Salvoda, McMaster, Canada, for her greetings and remarks on the behalf of the Heart Society, which deals with development, origins of health and diseases. I am thankful to Dr. Kate Kennedy from McMaster University, Canada, to deliver keynote lecture. Her lecture is valuable and thought-provoking for scholars from among global community. I am thankful to Ms. Nara Lopes, research scholar from Alberta University, US, Canada, for presenting excellent results of reaching importance, especially in present scenario. I am thankful to Professor Ruby Bhatia to deliver keynote lecture on safe motherhood and child health, which is so relevant for mothers from a very diversified socio-economic strata from a development country like India. I am thankful to Dr. Toby Mensah from Melbourne University, Australia, for delivering keynote lecture on tropical issues for contemporary times. His lecture imparted on many meaningful interactions by decent. I am thankful to webinar chair, Professor R.K. Bahal. Under his guidance and inspiration, we are being unable to organize this webinar. He is passionate researcher with human heart for fellow beings on global scale. I am thankful to Dr. Pooja Malik, hostess of today's webinar, for anchoring the proceeding of the webinar systematically and well in time. I would be falling in my duties if I missed to thank Professor Devi Singh, Professor M.K. Rana, Engineer Ramesh, Dr. N.K. Tiwari, Dr. Parveen Kumar Sharma, Dr. Ridhima Arya for painstaking effort to make this webinar great success. And thankful to all members of organizing committee and participants from different states in India and abroad to enhance the mission of webinar by their presence in the webinar as well as the guidance to organize. Once again, I thank one and all to make today's webinar successful. Real gift in respect of women in the society globally by celebrating International Women's Day. Jai Hind, Jai Bharat. Thank you.